Okay, so let's get started. So welcome back, everyone. So I'm Colin Palmer. I'm an assistant professor in the Department of Psychology, and I'm looking forward to teaching you about biological psychology this semester. Today I'm going to go through a general outline of the course, so give you a sense of the kind of topics that we will cover and what the assessments will involve and things like that. I'll take a few minutes to try to sell you on why it's interesting and important to learn about biological psychology. And then we'll go into the first lecture of the course, which will be a general introduction to the cells that make up our brain and our nervous system. So generally in these lectures we'll have a kind of like a 5 or 10 minute break midway through, but today's lecture is a nice short one, just to kind of help ease you into the new semester. And so I think we can probably go all the way through and then without a break and then finish early. Okay, so about me, so I came to Singapore 2 years ago to NUS. Previously I was in Australia, hence koala, and my background is in neuroscience and psychology. Most recently I was a researcher based in Sydney where I studied visual perception, and that's one of the topics that we'll cover in this course. Going back even further, I first became interested in biological psychology when I read these two books when I was sort of coming to the end of high school and deciding what to do at university. Some of you might recognize them. They're popular science books that describe, I guess, kind of peculiar cases in clinical neuroscience. And so this book, Phantoms in the Brain by V.S. Ramachandran, it describes this quite strange but surprisingly common experience that people can have after they've lost a limb, which is called phantom limb syndrome. And what happens is that a person has lost a limb, like they've been in war, they've been in a car accident or something like that, they've lost an arm or a leg, they've recovered from their injury and they're cognitively perfectly fine. But sometime after their injury, they start feeling sensations from the missing limb. So they know that their hand is not there anymore. They can see that it's not there anymore, but they still feel a sense of touch from that missing hand or a sense of pain. And it illustrates profoundly, I think, how our experience of the world around us, even something as personal as our experience of our own body, can be something that is just kind of generated or made up in our brain. So usually it will align with the reality of the world, but sometimes it can come apart from reality, what we experience. So in this course, the overarching goal is to give you an understanding of the relationship between biology and psychology, so between the brain and the mind. It's an introductory course, and so I won't be going into every kind of interesting topic in this field in detail, but I will give you a good grounding in the fundamental concepts of the field, and I'll go into a number of topics that I think are really interesting to learn about in a bit more detail. So for some of you, this will form the basis of further study in biological psychology, and so the psychology department offers a number of 4K modules that are on topics like memory and emotion, sleep, perception, consciousness, evolutionary psychology, and things like that. Where having a foundation in biological psychology is really important. But even for those of you who aren't interested in further study in this kind of field, I think if you're interested in psychology, then it's worth having an appreciation of what our brain is and what it's doing, how it shapes our behavior. Along the way, I will try to also point out things that we don't yet understand about how our brain functions and how it contributes to our mind and behavior. So biological psychology is, I think, one of the most exciting and active frontiers in science. There's been a huge amount of research over the past few decades in this field, motivated partly by the development of new technologies that allow us to peer into somebody's brain in real time and look at it functioning. We've learned a lot from that research, but there's still a number of really fundamental and interesting questions that are not yet resolved. And I think that's a part of what makes the field interesting, that there's still space here for new ideas to be developed in our understanding of how the brain functions. So these are the topics that we're going to cover. So the first 3 weeks is going to start at the very low level, the level of how individual cells in our brain function, what they're doing. And this will probably be the most technical part of the course, and it will involve learning about biology of cells, learning a little bit about chemistry. But it will be really foundational for understanding what our brain is actually doing and how it's possible for it to contribute to maybe more interesting cognitive functions, if you like emotion and memory and so on. In week 4, we'll focus on anatomy and also the research methods that are used in cognitive neuroscience to probe the relationship between the brain and the mind. Then we'll spend a few weeks talking about perception, so vision, hearing, smell, touch, and taste, and so on. And this is the area that I do research myself, and so I'm really looking forward to trying to convey what I find exciting about that field to you. And then for the rest of the course, we'll touch on emotion, memory, sleep and dreams, and genetics and evolution. So all of the lectures will be here in LT11 on Tuesday afternoons. They're face-to-face lectures, but they also will be recorded. And so generally the recordings will be posted on Canvas later in the week of each lecture. In addition to the recording, I'll also be putting up an edited transcript of each lecture online on Canvas after the lecture, so edited to make it more readable. And the purpose of that is mainly to help with your revision, so that all of the information in the lectures is written down for you somewhere. So during the lecture yourself, don't feel like you need to write down everything that I say. The tutorials are, as usual, sort of in-person small classes that run fortnightly. They begin in weeks 3 and 4. You're expected to attend all of the tutorials, and participation in the tutorials will contribute partly to your grade for the course. And we have a couple of quite experienced teaching assistants this semester, Melvin and Corinne, who you'll meet when the tutorials begin. The first 2 tutorials will be aimed at trying to help you consolidate and revise some of the more biological concepts from the first few lectures. So these will involve kind of hands-on activities that will hopefully give you a bit of a different perspective on the concepts that we cover in the lectures. In the second tutorial, there'll also be a bit of information about the midterm test. The third tutorial will be reserved to some time where you can get extra sort of consultation and feedback on the midterm test. And then later in the semester, there'll be a paper discussion on anxiety disorders and a tutorial aimed at helping prepare you to do well on the final exam. The textbook that I used for this course is called Biological Psychology. It's by James Calat. It's, I think, quite a good textbook. It's quite easy to read and interesting, well illustrated and so on. A number of the lectures, particularly earlier in the course, are based fairly closely on textbook chapters. Having said that, all the information that you need for the assessments will be covered in the lectures, and so if there's content in the textbook that I don't cover in the lectures, then you won't need to know it for the assessments. Having said that, the textbook will give you kind of a broader context than what I can cover in the lectures. It'll give you sort of different examples, and so if you're finding a particular topic that we covered in the lectures more difficult, a good place to start will be to read the textbook chapter. Okay, the assessments. So your final grade will be based 20% on tutorial participation, 30% on a midterm test, and 50% on a final exam. The midterm test will be in week 7. It'll be held during the lecture slot here in LT11 in week 7, so make sure that you're available for that time. It will be based off the lecture content that we cover in the first 6 weeks of the course. It will be multiple response questions, and there'll be more details on the format of the test in the second tutorial. And I'll also post a practice test on Canvas later in the semester, which will give you a clear idea of what the format will be like. The final exam will be based off the lecture content from the whole course from week 1 onwards. It will also include multiple response questions as well as some short answer questions where you need to write like a paragraph or something like that. And again, there'll be more details about the format of the exam in the tutorials. So I think most of you are very familiar with Exemplify and don't need any reminders about this, but I know that some of you won't have used it before, and maybe because you're doing exchange from overseas or something like that. And so both the midterm test and the final exam will be done using Exemplify. What that means is that you have to come to the test venue with your own laptop with Exemplify installed and the exam file downloaded. If you haven't used it before, you should brush up on it. So there's some links here where the university has some web pages with information about how to download Exemplify and get it working. So you should look into that before the midterm test in week 7. And the Central IT also offer student briefings throughout the semester that you can sign up for as well to learn about Exemplify. If you have any kind of technical issues with your laptop, or you don't have a laptop that you can use, then please contact CIT well before the midterm test to get their help with that. Okay, so if you have questions about the lecture content throughout the semester, my preference is that you come to the lectures and ask me in person. You can ask me during the lecture. You can come up to me at the end of the lecture and ask me. So I'll hang around at the end of each lecture. You can also post questions on the discussion tab in Canvas, where it will be viewable by all the students, and I'll get a notification when you do that and answer it on Canvas. If you email me with questions about the lecture content, I'll probably just ask you to post it on Canvas. And the reason for that is because I usually get similar questions from multiple students, and so it makes more sense just to answer them somewhere where everyone can see the answer and benefit from it. If you have more personal questions or kind of administrative questions that kind of just relate to you, then please send me a private message through Canvas. Okay, so speaking about Canvas, I posted this survey last week, and so thank you for all of you who took the time to fill it out. I've read through all of your kind of text responses and found it really useful to understand more about your background and what you're interested in regarding the course, what you're concerned about, and so on. I've just highlighted here a couple of the questions included in the survey. One was about whether you had taken previous courses on biology at university, and you can see that the vast majority, 75% or so, said no, and probably relatedly, there was a question about how difficult do you expect the course will be, and the majority said they expected it to be difficult. And so the reason I just wanted to show this briefly here is just to hopefully provide some reassurance that most of you are in the same boat here in terms of not having much background in biology, and also just to say that I'm aware of that, and I've designed the course with that in mind. Okay, so are there any questions at this point about the format of the course? Okay, so why study biological psychology? I think a short answer to that is just that biology, so meaning processes that are happening inside our brain and our body, genes that we inherit from our parents, and even our evolutionary history as a species, these things can help us to understand the function of our mind and human behavior. There are various different types of biological explanations that we use to understand psychological phenomena. So one type is a physiological explanation, which relates our behavior to the activity of our brain and our body. And so the idea that hormones, so chemicals released into our bloodstream, can affect the way that we feel, the emotions that we have, and the way that we react to events around us. This is a physiological explanation for our experience and our behavior. Another example, the idea that some people are more prone to getting kind of stuck in depression partly because of genes that they inherited from their family, is another kind of physiological explanation for their kind of life experience and their behavior. Another type of biological explanation is an evolutionary explanation that relates behavior to our evolutionary history as a species. So we'll talk a bit more later in the course about the idea that the reason that many of us are prone to experience anxiety in day-to-day life can be explained partly because this tendency to feel anxiety is and was useful throughout our evolutionary history, that it helped our ancestors to avoid danger, to survive and reproduce and pass down those kind of traits to us as well. If you've observed the macaque monkeys around Singapore, you might have been struck by how similar they are to humans in certain ways. So they get together into social groups. The juvenile monkeys play with each other. They kind of pester their parents in a similar way to how human children do. They communicate with each other using facial expressions. They seem to have emotional reactions like fear and surprise and aggression and so on. And one explanation for that similarity between the behavior of the macaque monkeys and human behavior is partly because we share a common ancestor, that there used to be, a long time ago, animals that had some of these types of traits that eventually evolved into humans on one side and macaque monkeys on another. Okay, so there are different kinds of explanations kind of different levels of explanation that we tend to appeal to try to understand human psychology. This figure here is meant to sum that up. So this is from something called the RDoC research framework, developed by the National Institute of Mental Health in the US. And this has been quite influential in guiding how research funding has been used in the US in kind of recent decades. And this figure is just illustrating a number of kind of different levels of biology that underlie human development and behavior. And so if we want to understand a particular psychological phenomenon like anxiety, we can do that at the level of behavior and self-report. We can ask a person how they feel. We can observe how their behavior changes. We can introspect on our own experiences of anxiety. We can also try to understand it at the level of physiology. So There might be particular hormones like cortisol that are released into the bloodstream when someone feels anxious. We may be able to identify particular circuits in our brain, particular networks of neurons that become active when someone feels anxious. And then we can go even sort of lower level to the level of cells and molecules and genes. So we might be able to identify particular genes that some people carry in their DNA and others don't, that make it more likely that they will experience an anxiety disorder during their lifetime. So to really understand anxiety, we need to be able to explain it across these different levels of description and understand how these different levels interact with each other. Okay, so in terms of why it's sort of useful to study biological psychology, one thing that where I found it useful is understanding new research findings, where neuroscience is kind of more and more related to how we understand human psychology. And so one example is if you kind of read an article in the news that's just come out that says a new study discovered that a mindfulness intervention increases brain activity in the dorsolateral prefrontal cortex, there's a question of what does that actually mean? Is that a good thing? Is it a bad thing? Is it completely irrelevant for our understanding of mindfulness? And to know that, you need to know something about what the dorsolateral prefrontal cortex is, where it sits in relation to the rest of the brain. You need to know something about what brain activity actually is. Like, what does it really mean for the brain to become more active? And how does that contribute to our understanding of mindfulness? This course should also give you a better idea of how drugs interact with the brain, including psychiatric medications, how it's possible for chemicals that we ingest to influence our experiences and our mood and our behavior, why side effects occur, how newly developed psychiatric drugs might work better than those that have been available to us in the past. Some of you might be interested in how this field relates to new technologies, and so there's a lot of interest in AI at the moment, and a number of developments in AI have been driven by things that we've learnt about how the brain processes information and how the human brain develops and kind of organises itself. Another example is VR, or virtual reality. So if you've had the chance to try out sort of a modern VR headset, you'll know that these can produce very compelling and realistic experiences of being located in a 3D virtual environment. And the development of that technology has really been driven by what we understand about how the brain produces our experience of the world around us. And then finally, I think that biological psychology is just important for understanding more about kind of who and what we are as humans. So I hope that you'll come away from this course just feeling like you've learned something interesting about yourself in terms of processes going on inside of your brain and inside your body that help to shape your kind of everyday experiences. Okay. Okay, so now we can go into the first lecture of the course, and this will be a general introduction to the cells that make up our brain and make up our nervous system. So to understand how our brain functions and how it can contribute to our psychology, we really first need to understand what the individual components of our brain are doing. By the end of the lecture, you should be able to describe the two main types of cells that make up our nervous system and describe the basic structure and function of one of these cells, which is called the neuron. We can start with a general overview of the nervous system, and it's often kind of divided into what we call the central nervous system, which is made up of our brain and our spinal cord. and the peripheral nervous system, which is shown in blue here, which refers to all the other nerves that run throughout the rest of our body. If we zoom in on the brain, the human brain at the bottom here, you can see straight away that there are clearly different structures that make up our brain. We can see that with the naked eye, if you like. And some of those are labelled here. So we have the cerebral cortex, or just the cortex for short, which is this wrinkly structure, which is kind of making up most of what we can see from this view outside of the brain. We have the cerebellum at the bottom here, this structure that sits at the kind of base of our head at the back. And then we have the brainstem, which is the part of the brain here that connects the brain to the spinal cord. And if we look at the brains of other species, in particular other mammals, what we find is that a lot of these structures are there as well in those other species, okay? So the brain of a monkey and cats will also contain a cerebral cortex, a cerebellum, a brainstem, and so on. So these are not uniquely human structures. So what does our brain actually consist of? An important piece of history in neuroscience and psychology is the discovery of the neuron. And so the neuron is the main type of cell that is found in our brain that we'll be focusing on in this course because of its role in cognition and psychology. And the neuron was first described by a Spanish researcher named Santiago Ramon y Cajal. And he was looking at brain tissue through a microscope, and a new technological development that allowed him to see the neuron for the first time was something called a staining technique, which is where a particular kind of chemical is applied to a piece of brain tissue, and that chemical stains, it changes the colour of a small subset of the cells that are present in that brain tissue, and that makes the individual structure of those cells easier to discern. And so on the left here, we have an image of a neuron as seen through a microscope. The neuron has been stained a darker colour compared to the rest of the brain tissue around it. And because of that, it's easier to see that kind of individual structure that makes up this particular cell. And so this was a really important discovery, being able to actually see an individual neuron for the first time, partly because it showed that our brain really is made up of these individual components that have a particular structure to them. So it's common knowledge nowadays that our brain is made up of brain cells, that just like every other organ in our body, it's made up of these tiny little components that we call cells. But at this time, it was actually unclear and debated until we had the technology to see those components directly. And so Cajal won a Nobel Prize in the early 1900s for his work. This is a drawing that Cajal made of some of the neurons that he saw through his microscope. And you can see immediately that there's diversity here, that different neurons can be different sizes, they can be different shapes, and so on. But we'll see in a moment that despite that diversity, there's still a common basic structure that most neurons have, which relates to the way that they function. But first of all, it's worth, I think, trying to illustrate the complexity of what we're dealing with when we try to understand the brain. And one question we can ask is how many of these components do we have? How many neurons do we have? And recent estimates are putting this at something like 86 billion neurons on average. And so the majority of those neurons are actually found in the cerebellum, that structure at the back of our brain, which is very densely packed with neurons. The cerebral cortex, which we'll be talking about a lot during this course because of its importance to cognition, has something like 16 billion neurons, and this area of our brain occupies the largest volume of our brain. And then even our spinal cord has something like 1 billion neurons on average. And so that number of 86 billion it's a really large number. And we'll see in a moment that what these neurons are engaged in, which seems to underlie our cognition, is communicating with each other, sending signals to each other. And so what that means is that right now in your brain there are billions of these individual cells that are all actively involved in sending signals to each other. And it's that activity of this hugely complex and interconnected system that is able to produce your thoughts and your feelings and your experience of the world. To further illustrate the complexity of the brain, I thought it would be useful to tell you a little bit about a particular study that I saw at a conference a few years ago, which developed a new technique for visualizing the brain. And so, in particular, what they did is build a 3D model of all of the neurons that are present in a particular area of brain tissue. So to do that, they extracted a very small block of brain tissue from the cortex of a mouse, and the block of brain tissue is illustrated here, and you can see that it's measured in, its dimensions are in micrometers, so a small fraction of a millimeter. And then they used something called an electron microscope to create to capture a cross-sectional image of sort of one end of that block, which is kind of shown here. And what you're seeing in that image is just sort of structures of the cell, like the boundaries of different cells that are all sort of bunched together. And then they scraped off a very fine part of that end of the block of tissue. They captured another cross-sectional image of the block, and then they repeated that process until they'd worked their way through the whole block of tissue and had a set of two-dimensional cross-sectional images, which they could then use to reconstruct a three-dimensional model of all of the neurons that are present in that brain tissue, which is what's shown on the right here. So here, this video is just highlighting individual neurons, one at a time, that are found inside that block of tissue. And you can see that each of them is sort of sending out these extensions that sort of tangle up with the other neurons that are present. This is another view of their 3D model. And so the camera here is this kind of tunnel that the camera is flying through is actually the space that's left by a blood vessel. And all of what you see here, what's included in the model, are just the neurons. Okay, so all these neurons are sending out these kind of wires or extensions that are all sort of intertangled with each other and forming this very complex interconnected network. Okay, so most of the course will focus on neurons, but it's also worth, when we're talking about the cells of the brain, I mean, but it's also worth mentioning that there is another main type of cell present in our nervous system, and that is the glia cell. And so there are diverse types of glia cells, so some of which are illustrated here. But generally, they each have different types of functions in our brain, but generally they play a role in supporting the function of neurons. And so these glia cells are also very prevalent in our brain. In some areas of the brain, they outnumber neurons, but generally it's the neurons that seem to have a more direct role in driving our cognition and behavior. So, but just to give you a feel for what glia are doing, one example shown here is called an oligodendrocyte, and we'll actually mention this again in next week's lecture. And this glia cell has the role of insulating neurons. So it actually wraps itself around the neurons, and that insulation allows the neurons to send signals more efficiently. Another example are microglia, which have a role in our immune system, and so they're involved in fighting off pathogens that enter our brain. And then one more example are the astrocytes that have a few different functions, but one being to regulate how nutrients are being transferred from our blood vessels to our neurons. Okay, so the main point here just being that these glia have different roles in supporting the function of neurons. So the key learning points are that the nervous system is composed of two types of cells, the neurons and the glia, and that our brain contains many billions of these neurons. Okay, so now we can look at the structure of a neuron in more detail. And generally, neurons have three main components to them. They have a cell body, which is pictured here. Sometimes you might see it referred to as a soma as well. And this is the central kind of body of the cell that contains the nucleus of the cell. Neurons also contain dendrites, which are these protrusions that extend out of the cell body and tend to look like the branches of a tree. And these dendrites have a really specific functional role, which is to collect or to receive signals from other neurons and then transmit those signals to the cell body of the neuron. And then finally, neurons also have an axon, which is pictured here, this longer extension that comes out of the cell body and has this different function to the dendrites, and that is to send signals to either other neurons or to other structures in our body, like our organs or our muscles. And so some of the features of a neuron are shared with other cells in our body. So almost every cell in our body has a nucleus that contains a copy of our DNA. But the really sort of more characteristic and defining features of neurons here are the dendrites and the axons, which are specialized to send and receive signals with other neurons. This is an image of a real neuron seen through a microscope, and you can fairly easily identify these main structures. We have a cell body, we have dendrites that are branching out from the cell body, and the axon is sometimes difficult to distinguish from the dendrites just from sight alone, but here is this longer extension coming away from the cell body. So then what are neurons actually doing? And it turns out that the main function that neurons seem to be engaged in our brain, and particularly related to cognition and behavior, is in communicating with each other, so sending signals to each other. This includes an electrical signal called the action potential that we'll talk more about in next week's lecture. So this is an electrical signal that's generated at one end of the axon near to the cell body and then travels down the length of the neuron's axon. And when it gets to the end of the axon, it triggers a chemical signal to be released from the axon of the neuron. And that chemical signal, which involves the release of chemicals called neurotransmitters, can then be detected by often the dendrites, sometimes the cell body of another cell, and in that way a signal is then passed from neuron A to neuron B. So these electrical and chemical signals are really important for understanding our brain, because it's by communicating in this way and forming together into networks that are able to process these signals in different ways, that neurons are able to effectively process information to control our body, our behavior, including the movement of our muscles, and also to receive information from the outside world, from our senses. And so all features of cognition, like memory, emotion, decision-making, and so on, are likely to rely in some way on this communication happening between neurons in our brain. And we'll see many examples of that throughout the course. This process of communication between neurons is also important for understanding things like how medication can affect our brain, how it's possible for chemicals to influence our brain in a way that affects our behavior and our experience, as well as how our genes that we inherit can affect the development and the function of our brain. And so for the next couple of weeks of the course, we'll focus on first the electrical and then the chemical signal in some detail, and that will be an important foundation in being able to relate the activity of our brain cells to cognitive functions. Okay, so a term that you might have heard before is the synapse, and exactly what is this? It's a point of connection between two neurons, and so where the axon of one neuron comes very close to the dendrite or sometimes the cell body of another neuron, it forms a synapse. So the synapse is a specialized kind of junction point between neurons where signals are transmitted. One feature of synapses is that the communication between neurons that happens at the synapse is mostly one way, okay? So in this example, neuron A is sending a signal across this synapse while neuron B is receiving that signal, but generally not sending a signal back to neuron A. And so because of that one-way communication that occurs at a specific synapse, we can define for that synapse one of the neurons as being the presynaptic neuron, the one that's sending the signals, and the other neuron as being the postsynaptic neuron, the one that is receiving that signal. And so that terminology will be useful in the following lectures. Okay, so this is a real synapse, so pictured using an electron microscope, which is a type of microscope that can produce very high resolution images, including of cellular structure. What you're looking at here is at the top, we have the axon, the end of the axon, which is also sometimes called the axon terminal of the presynaptic neuron. At the bottom, we have a part of the dendrite of the postsynaptic neuron. And then in between, we have this tiny gap, which is called the synaptic cleft. So it's across that gap that chemicals can be released from the presynaptic neuron and then detected by the postsynaptic neuron. One other structure of neurons that I'll mention just briefly is something called dendritic spines. And these are tiny little protrusions that extend along the length of the dendrites. And so what we're looking at here on the left is a neuron highlighted in white. We're zooming in on a part of its dendrites in panel B here, and you can see that there are all these little protrusions that kind of stud the length of the dendrite. So these are the dendritic spines. And it's at these spines that synapses are usually forming. So what's not shown here is that there will be axons from many other neurons that are extending out and forming synapses with the dendrite of this neuron that's highlighted in white, and most of those synapses will be occurring at the dendritic spines. Okay, so most of what I've talked about so far is the case where one neuron is sending signals to another neuron, and out of the many billions of neurons in our brain, this is what most of them are engaged in. But neurons can also be specialized for other functions. So one example is what we call sensory neurons, which are specialized to receive signals from our senses. So our eyes, our ears, our skin, and so on. In this example, this is a sensory neuron that is involved in detecting touch on our skin. And so what you're looking at here is what are called sensory endings of this neuron, which are similar to dendrites that are embedded in the skin. When touch occurs on the skin, it will cause a signal to be generated in those sensory endings, an electrical signal that travels down the length of the sensory neuron, down its axon, to where the axon forms synapses with other neurons that might be located in our spinal cord or in our brain. And so in this way, information about touch on our skin is being transmitted from the periphery of our body into our central nervous system. So that's a sensory neuron. Another example of a specialized type of neuron is a motor neuron. And this refers to neurons that are involved in directly controlling the contraction of our muscles. And so in this schematic, what you're looking at is a neuron that is extending its axon out to form synapses with a muscle fiber. And when this neuron is active, an electrical signal will travel down its axon and stimulate the release of neurotransmitter onto the muscle fiber and cause it to contract and cause our body to move. So this is what's happening every time we move. That there are, so right now as I kind of flap my hand about, there are neurons in a region of the brain called the motor cortex, which is located at the top of our head. These neurons have their cell bodies and their dendrites located there in the brain, but their axon extends from that region of the brain down into the body, into the spinal cord. And so electrical signals that are generated in that motor neuron travel down into the spinal cord and then are relayed out to the muscles of my arm and cause the muscle fibers to contract and my body to move. So these are motor neurons. Okay, some basic definitions that are worth mentioning. One is the idea of afferent signals, which refers to signals in our nervous system that are carrying information into our brain or into our central nervous system. And so sensory neurons are a key example of neurons that send afferent signals. Okay, so their cell body will usually be located somewhere out in the periphery of our body, like in our arm, for instance. But they're carrying information along their axon into our spinal cord, into our brain. So this is an afferent signal. This contrasts with efferent signals, which are signals in our nervous system that carry information out of our brain or out of our central nervous system into the periphery of our body to influence the function of our organs or our muscles. And so a key example of this is motor neurons, where signals are being transmitted from our brain out to control our body. And then these also contrast with what we call interneurons, which are neurons that are carrying signals within a particular area of our brain between two other neurons. Okay, and so interneurons might be located in a specific part of the brain. Their dendrites will be there, their axon will be there, and they're just carrying signals across that local area rather than sort of out of our brain or into our brain. Okay, and so the significance of these signals that neurons are sending to each other becomes apparent when you look at how neurons are organized into networks or into circuits. So generally the neurons in our brain and body are organized into circuits that are able to achieve some particular function. And so to give you a concrete example of that, if we consider a really simple behavior, which is what we can call a motor reflex, where if you imagine that you're cooking dinner, you're frying some dumplings in the frying pan, and some hot oil jumps out of the pan, lands on your hand, and you react by quickly pulling your hand away. So that very simple behavioral response can be also explained in terms of a very simple neural circuit, which is illustrated here. And so to unpack what we're looking at here, first we have a sensory neuron that has its sensory endings in our skin. And when that hot oil lands on our skin, the temperature or the damage it does to our skin will trigger a signal to be generated in the sensory endings of the neuron that is then transmitted down the length of its neuron, down to where its axon forms a synapse with another neuron that's located inside of our spinal cord. Okay, so a signal conveying that event, that touch on our skin, is being conveyed into our spinal cord, and the axon of the sensory neuron in this example is forming a synapse with the dendrites of a interneuron that's located inside of our spinal cord. So the signal is then passed on by that interneuron to where it forms a synapse with the dendrites of a motor neuron, and then that will generate a signal in the motor neuron that will then pass down the length of its axon that extends out of the spinal cord down into our body to come in contact with the muscles in our arm, for instance, and trigger the muscles to contract and pull away from the hot oil. Okay, so the idea here is just that behaviors can sometimes be understood in terms of circuits, so the way that neurons are connected up to each other. And we'll see some examples of how, when we get to the brain, neurons form into much more complicated circuits that are able to process information in more sophisticated ways and allow for the more flexible control of behavior. Okay, so the key learning points here are that neurons have a cell body, they have dendrites that are specialized to receive signals from other neurons, and they have axons that are specialized to send signals to other neurons or to other structures. Neurons communicate with each other using these structures called synapses, these specialized points of connection between the axon of one neuron and the dendrite, or sometimes cell body, of another neuron. And it's at these synapses that chemical signals are sent or released by the presynaptic cell and detected by the postsynaptic cell. And then neurons can also be specialized to receive signals from our sensory organs or to send signals to our muscles. Okay, so are there any questions at this point? Okay, so to finish up the session, we can do a few multiple choice questions, which we're going to do through Poll Everywhere. So I'll activate the question in a moment, but you can access it through the link here if you've got the slides on your computer, or you can scan the QR code with your phone. And so these questions are not kind of graded in any way. You can put in an answer anonymously. The idea with it, and I'll be including these questions in each of the lectures, is to give you a bit of feedback on whether you're following along with some of the key concepts from the lecture. Give me feedback as well. And sometimes I'll use these questions not really to evaluate your understanding or anything like that, but just to help illustrate a particular concept that I want to illustrate in more detail, or just to kind of serve as a pretext to kind of talk a bit more about some of the concepts. Okay. All right, so the first question here is to imagine a neuron that has a cell body located in your left leg and an axon that extends from your left leg into your spinal cord. And the question is, what is a plausible function of this neuron? A, is it to help transmit signals from your brain to control your muscles? B, could it be to help transmit information about touch on your skin towards your brain? Or C, does it depend on whether this is a sensory neuron or a motor neuron? So I'll give you a moment to think about that and put in an answer. And of course, feel free to chat to the person next to you about the question. Okay, so it looks like I think most of you have put in a response already. So so 80% or so put option B, and a fair few also put option C. And so that the idea of this question is that it relates to these different functional roles that axons and dendrites have in neurons. That we know that the dendrites connected to the cell body are receiving signals, whereas the axon is extending away from the cell body, sending a signal to somewhere else. And so if the cell body is located in our left leg and the axon is extending from there into our spinal cord, it implies a particular direction that the signals are being sent, that they're being sent from our leg to our spinal cord. And so B is the correct answer, that a plausible function is to help transmit information about touch from our leg into our central nervous system. C is it's correct that the role of neuron depends on whether it's a sensory or a motor neuron. But in this case, what we've learnt about this neuron in terms of where its cell body is located and where its axon is extending to already implies that it can't be a motor neuron, which will be one that's sending information from our brain to our muscles. Okay. All right. So another question is, how do glia differ from neurons? A, neurons make up the central nervous system, while glia are found mainly outside the central nervous system. B, there are diverse types of glia, but only one type of neuron. C, glia are few in number compared to neurons. Or D, glia often play a role in supporting the function of neurons. Okay, so pretty much everyone put D, which is correct, so great job. So we talked about how glia have these different kind of diverse roles in supporting the function of neurons. Both glia and, so A isn't correct because both glia and neurons are found sort of both in our periphery and in our brain, in our central nervous system. There are diverse types of glia, but we've also seen in this lecture that we can also define some different types of neurons as well. So that's why B isn't correct. So sensory neurons and motor neurons, for example. And I mentioned as an aside that glia are also quite numerous in our brain, but also if you know that they're playing a role in supporting the function of neuron, it's likely that they'll also be in quite prevalent across our brain and our body. Okay, so question 3. The question is, how does the axon of a neuron differ from the dendrites? A, the axon transmits signals away from the cell body, while dendrites collect signals that are passed towards the cell body. B, the axon receives signals from other neurons, while dendrites transmit signals to other neurons. C, the dendrites are usually responsible for processing sensory information, while axons usually control movement. Or D, the axon and dendrites have some structural differences, but they appear to serve a similar function. Okay, so that seems to be a pretty straightforward one for the class as well. So almost everyone chose A, and again, that's the correct answer. We've talked about these different functional roles that the axons and dendrites have in communicating with other neurons. Yeah, and so C, for example, isn't quite correct because neurons are involved in both in processing sensory information. Sensory neurons and neurons that control movement will have both, typically often have both sort of dendrites and axons. So yeah, so that isn't quite correct. Okay, and then one final question. It's about the number of neurons in our nervous system. So approximately how many of these neurons do we each tend to have? Is it 8,600, 86,000, 86 million, or 86 billion? Okay, so yeah, so everybody pretty much got the right answer again. So the answer is D. So just the concept or the point that I wanted to get across is just that the number of these neurons in our body is in the order of billions. It's a very, very large number. Okay. So with these poll everywhere questions, I usually forget to mention this until we're kind of deep into the course, but I have put the correct answers on the last slide in small text, so you can always sort of look that up as well. And yeah, that's it for today's lecture. If you have any kind of outstanding questions, you're welcome to come up and ask me now. But otherwise, I'll see you next week when we'll talk about these electrical signals that neurons send in more detail. So thank you. See you next week.